Good morning to everybody. It's good morning in Los Angeles, and obviously around the world, it's a different time. I'm Jeffrey G. I'm the Executive Director of the NDF, and I'm delighted to welcome you aboard. I am told that we have people on the call from Japan and from India, and I'm sure from many other countries around the world. We are very much honored today to have with us Professor Stella Mitrani-Rosenbaum. And I'm sure each of you have seen her bio, know exactly who she is. She's joining us today from Hadassah Hospital, Hadassah Medical Center in Jerusalem, and she's going to give a talk on the work that she's been doing. She has been so much involved in everything that NDF has done. More than 20 years ago, she discovered the gene that causes GNEM. She has been working as a scientific advisor to NDF for many, many years, and we are highly honored that she is part of our team. Also joining us today is Dr. Noah Weisleader. Noah serves on our scientific advisory committee, and here again is one of our esteemed scientific colleagues, and we are very pleased to welcome them both. So, Professor, I will leave the floor to you. I'm here if needed, and we look forward to your talk. Thank you.
Hello, everyone. Thank you for joining us on to hear our developing animal model talk. So we will hear today an update of our work on animal models for GNE myopathy. We will hear first on the zebrafish GNE knockout model that we have established and already characterized. And we will also hear about the mouse GNE knockout model that we have completed now, but we are now beginning to characterize it. And finally, in the last few minutes of the talk, I would like to introduce you to the ProDGne European Consortium. That is an European team that is also working on GNE myopathy. Okay, so we all know GNE myopathy, unfortunately. We all know that GNE is the gene causing the disease. And we also know the function of the gene, or the non-function of the gene at least, that is the catalyzation of the first two steps in sialic acid biosynthesis. But all this hasn't led us to a disease treatment yet, unfortunately. So what do we need to have a disease treatment? First, of course, we need to know the etiology of the disease, then the mechanism of the disease. Then if we know that, we can design a treatment according to the etiology and the mechanism. And then, of course, we need a biological model to assess this mechanism and to assess the efficacy of any treatment that we have designed. So on the etiology, as I said, we all agree mutations in the GNE gene are responsible for the disease. On the mechanism, this is a little bit more controversial. Most researchers, most people think that the mechanism of the disease is a sialic acid deficiency, but maybe there are also other mechanisms involved in GNE myopathy that are based on additional GNE functions, for example. Then the treatment designs that have been done today are based, of course, on the sialic acid deficiency hypothesis first, but also with a more comprehensive strategy that is gene therapy. And finally, the biological model to assess these treatments, here we have a little problem still, but hopefully we will also overcome it. So what are the models that we can use today? So we have some cellular models that are mostly including cultures of muscle cells from patients, but this characterization and also from biopsies of the patients showed very mild effects of the GNE mutations. So we couldn't not point to a very definitive mechanism for a GNE myopathy in those cells. Then at the level of animals, we don't have any informative animal model yet. We know that the knockout of the gene is lethal at the embryonic stage. We know that knock-in of different mutations does not recapitulate GNE myopathy features. And we know that a transgenic model that seemed to work at the beginning is not relevant anymore. So we do not have animal models to learn about the mechanism and to assess any therapy. So we thought that we should have another strategy, and that is to explore systems where the effect of the GNE mutation is more dramatic than the mutations that we know from the patients. So for that, we thought we should knock out the GNE gene completely. So the lack of the protein would really lead to some more dramatic effects on cells or on animals. So what we have done is to build cell lineages that are GNE knockout. And this we talked about last year, I think, about that. We have a muscle GNE knockout cell lineage that we have characterized and is now, hopefully it will be published very soon. But today I want to talk to you about our in vivo models. That is a model in the zebrafish and in the mouse. So let's begin with the zebrafish. These studies have been done in the lab by Hagai and Alon. They are based on the very strong conservation of GNE, specifically here between human GNE, between the human and the zebrafish. You can see that we have exactly the same structure of the gene, the same functions, and also the most of the mutations, and here we show only what we call the Middle Eastern mutation, are conserved in the zebrafish gene. So what we did is to knock out the GNE gene in the zebrafish by the CRISPR-Cas methodology. And after many manipulations and many design strategies, we target exon 3 of the gene, because we know that if we knock out exon 3, all the protein is knocked out from other systems. And indeed, finally, we got a fish which had a 4 base pair deletion in exon 3 that lead to a stop codon very near. So we have now a very truncated protein, a very small protein, 1 to 5 amino acids versus the wild type that is 725. And most likely we don't have this protein at all because it is degenerated. So this is the zebrafish with no GNE in its genome. And what we can see is that these zebrafish develop completely normally, at least up to 7 days after fertilization. Here we have a GNE wild type fish at 7 days post-fertilization. And here we have a GNE knockout fish. You see that they are completely the same. But then after these 7 days, around 8-9 days, we can see a phenotype that is appearing in GNE knockout. Again, this is the wild type fish at 8 days post-fertilization. And here at 7.5 or 8.5 days, we can see that the fish are beginning to curve. Okay, their body is now not straight, but curved. And also here, the swim bladder is getting completely deflated. Now, what it's most striking in this GNE knockout fish at 8 days, that 24 hours after the appearance of this phenotype, the fish simply die, okay? So they develop normally, then for about 24 hours, they show a phenotype, and then they die. So that is quite striking. Indeed, if we look at the motility of this fish, we can see here the white-eyed fish that swims very freely. Here we can see the knockout fish that is on the side. Okay, let me see again, you can see it. Here it's swimming on the side, it cannot be straight, and it swims very, very slowly. Also, if we look at what we call a touch-evoked test, that is, we touch the fish with some pipette, and we see the reaction time of this fish. So we see that at 8 days, the normal fish responds very strikingly, very rapidly. Here, the knockout at 8 days seems to be quite sick, but he responds to the touch evoke stimuli. And here at 8.5 days, the fish seems to be almost dead, right? It doesn't respond at all, but in fact, it is not dead. We can see the heart of the fish. You can see the heart beating, I believe. Can you see that? Yeah. So this is the normal rhythm of the white-eyed fish that's beating. And here we see the knockout fish. You can see that the rate of the beat is much, much slower. Okay, you can see it takes a long time for the heart to beat. And indeed, if we quantify this, we can see that at 5 or 6 days, there is no difference in the heart rate between the white type and the knockout fish. But then at 7 days, there is a decline that is beginning. And then at 8.5 days, we have a very dramatic difference in the beat rate of the fish. So we have this fish that develop normally and die almost suddenly at 8-10 days. And we think that we thought that we could maybe check in this system whether indeed sialic acid is responsible for all these phenotype. I mean, the lack of sialic acid is responsible for this phenotype. Again, we know that GNE is a key enzyme in the production of sialic acid, and the hypothesis, one of the hypotheses now is that GNE myopathy is caused by a lack of sialic acid. So this was a good system to check that, at least in fish, to see GNE knockout lack of sialic acid leads to the phenotype and to the death of the fish. So we took heterozygous fish, we crossed them to get the embryos at 1 cell stage, so it's just when they are delivered from the body. So we take them in a petri dish with normal water for them to develop, or in water where we have added sialic acid at different concentrations. These are the concentrations known to penetrate zebrafish, and we know also that in other sialic acid diseases, some could be rescued by those concentrations. So then we have this fish in the petri dish for about 16 days to be sure that we get everything there, because in those petri dish we have also normal fish, white-eyed fish, heterozygote fish, and also the knockout fish. Okay? These are the descendants of this crossover here. So we don't know ahead who is who. So we get them for 16 days. And then finally, after 16 days, we have collected all the fish also the ones that had died during this period, and we have genotyped them. And what we can see that the GNE knockout fish with sialic acid have not been rescued. The phenotype cannot be rescued by sialic acid. We see exactly the same phenotype. And most importantly, all the GNE knockout fish that were in sialic acid died exactly at the same time, that is 8-10 days after fertilization, meaning that sialic acid in this constellation at least could not rescue the phenotype nor the longevity of the fish. So now we wanted to look at the muscle of the fish to see if the GNE knockout really affects muscle. That's why we did that, right? So we used an assay called the Birefringence assay. This assay can visualize the light refraction of a very organized tissue. Here, for example, this is a white-type fish. By Birefringence, we can see the very well-organized muscle organization here. And if we look at different muscular dystrophy models that have been studied before, we can see indeed here a defect in the birefringence. You can see here there are regions of the muscles that do not refract the light as good as the white type, also for Duchenne muscular dystrophy. And these points indicates that there is some defect in the organization of the muscle. So we wanted to look at that with our fish also. And indeed, at 6 days post-fertilization, we see no difference in the birefringence assay between the wild type and the knockout fish. So here, the muscle tissue seems to be very well organized. Then we looked a little later, and indeed at 7 days, we begin to see some decrease in the birefringence assay in the knockout fish versus the wild type. And then at 8 days post-fertilization, we see a real difference in the birefringence between the knockout and the wild type. So again, this indicates that the muscle of those fish have some disorganization. To have more insight in this, we did muscle stainings of the entire embryos with anti-myosin, so to look at the myosin of the fish, of the muscles. Here we see a GNE wild type. We can see the very regular organization of the muscle fibers in this wild type fish. When we look at our GNE knockout fish, we can see here a real impairment, curvy shape of the fibers, maybe even fibers are missing. So indeed, we think that there is an effect of GNE knockout on the muscle organization of the fish. Then we did histology of those fish. That means we did cross sections of the fish, and we can see here again a GNE wild type fish. Maybe we can look at this right panel here. We can see the fibers of this wild type fish. We can see some compartmentalization of the different fibers. Okay, we can see that. And if we look at our fish, the GNE knockout fish, here below, you look at here at the right panel again, you can see that there is a completely lack of compartmentalization. We see the fibrils all across the muscle, but we do not see an organization in muscle fibers like here, okay? So here we can see really that there is a severe impairment of muscle fiber organization in the GNE knockout cells. And this occurs already at 5 days post-fertilization, so that is before we can see any of the phenotype, of the macroscopic phenotype of the fish. So we have seen here in this system that GNE is an essential gene in zebrafish, as in mammals, that is, with no GNE, the fish cannot live, and they die between the 8 and 10 days post-fertilization. And we can also see that the phenotype appears just before they die. And this phenotype includes decreased motility and defects in muscle fiber organization. So to have more insights and try to understand better the mechanism of the GNE knockout zebrafish phenotype, we did RNA sequencing on those fish. And we could see that at the different time points of their lives, 3 days, 7 days, 5 days, there are many differentially expressed genes. Although at 3 days post-fertilization, very few genes are different in those fish, between those fish and the wild-type fish, only 6. At 5 days, a little more, around 100, and then around 1,000 at 7 days post-fertilization. Then if we tried to organize those different expressed genes in pathways, and what we really see is what we knew from other, for other systems with GNE myopathy or GNE mutations, that the sarcomere organization is affected. Also the protein ubiquitination pathway. But what is really novel for us here is that cell cycle control, cell death and survival, and DNA damage and repair pathways are impaired in the GNE knockout cells. Sorry, in the GNE knockout fish. And this is very interesting because in the GNE knockout cells that I mentioned before, the system we have already established, we can also see these pathways coming up from the differential expressed genes. So we think that we have a GNE knockout zebrafish model that is to date the only animal model presenting clear muscle defects, thus possibly relevant to GNE myopathy. And therefore, we think that this model can be used as a platform for large-scale screening of potential drugs or different treatments to rescue the muscle pathogenesis or even the longevity or the life of the fish. And finally, the novel pathways that we can detect here reveal new clues on GNE functions, and of course, these have to be studied much more in depth. So now I want to talk to you about the development of a mouse model. So as I told you before, we are developing a mouse conditional GNE knockout model to evaluate GNE function in muscle. This work has been done mostly by Avi and Lena in the lab. And why that? Because again, we do not have very good GNE mouse models. We know that mice carrying GNE myopathy mutations do not show muscle pathology. We know that GNE knockout mice die at the embryonic stage, most likely from brain damage. So we cannot learn anything on the effect of lack of GNE on muscle in this model. So what we wanted to do is to establish a GNE knockout mouse, but that the knockout would be only in muscle tissue. So those mice will be born and will develop, and we will follow the muscle tissue development. So for that, we used as a platform a Gne-Flag mouse that we established before. That is a mouse with a tag at the very end of GNE, that is part, in fact, of GNE. And this is because we do not have, till today, a good antibody against GNE that can detect GNE in tissues of organisms, of mouse or of humans. So it's difficult to follow the GNE protein. So we have this flag GNE, and there is a very good anti-flag antibody. So GNE-Flag can be recognized in the tissues by this antibody. So we have used this mouse as a platform to establish our conditional GNE knockout model. So we have taken the strategy as follows. We have to introduce in the DNA of these mice 2 sequences that are called LOXP-T, flanking the gene that we want to knock out, so GNE for us. Then we use an enzyme, a recombinase enzyme that can recognize these sequences. And when it recognizes them, it cuts them like scissors and, in fact, excise what is in between. Okay? So we wanted to introduce the LOXP sequences flanking the GNE gene, then use this recombinase enzyme to cleave the GNE gene, but we, in fact, don't want that the recombinase will work on all the genes from all GNEs from all the tissues. We want it to work only in muscle, okay? So for that, we use a CRE that is led by a promoter that is specific to muscle, so that CRE will be expressed only in muscle tissue and not in other tissues. Okay, so once we will have these mice with LOXP sequences around GNE, we will cross it with a mouse that has a CRE enzyme led by a muscle-specific promoter. So we will get mice that have both the LOXP gene, the LOXP sequences, and the CRE. Okay, then we will, then this CRE will recognize the LOXP sequences only in muscle and excise the gene. Okay, so that's the strategy. But in fact, we have added a little tip here also. We have used a specific modified CRE that doesn't work automatically when it is in muscle, but only if we inject tamoxifen. So when we have these mice, they are completely normal. CRE doesn't work, doesn't recognize the LOXP sites. Only when we inject tamoxifen, CRE can recognize the sequences and excise the GNE gene. Okay, so this was the strategy, and it worked. So what we did was, again, to target exon 3 from GNE, to add at both sides of exon 3 the LOXP sequences. Again, we did that by CRISPR-Cas. We wanted to do that both at a time, that is to put in everything in one embryo or many embryos, and hope for the best, that is that we will have descendants with both the LOXP sites in the same allele of GNE. Okay, so we manipulated more than 400 zygotes to get the whole thing inside them. And what we got finally were 80 pups that were born. But what we found that we had 6 that had the LOXP2 sequence, 2 that had the LOXP3 sequence, but unfortunately none that had both sequences at the side. So what we did is that we established a colony of mice that had only the LOXP2 at the side, and we wanted to use them after that to introduce the LOXP3 at the other side, okay? So we had to generate a very big colony of GNE-Flag with LOX2P. In fact, we used more than 1,000 zygotes that were manipulated to introduce the LOXP3, okay? So from these 1,000 zygotes, 250 pups were born, 45 had the LOXP3 at intron 3, and from all those, after sequencing and very thoroughly looking at the exact sequence, we found 5 founders that carry the intact LOX site on intron 3. So now we have an heterozygote mice with both LOX flanking exon 3. We cross them with a mouse that is commercially available, so we just bought it. We didn't have to establish the lineage. We bought it from Jackson Labs. So we have the CRE, modified CRE, with alpha-actin promoter that is specific for muscle. From this cross, we get heterozygote mice that have also the CRE transgene. So now we have to cross them again with the original LOX mice to get finally homozygous mouse that have the LOX sequences in both alleles and have the CRE gene also in there, okay? So we have those mice, and we wanted to look indeed if by injecting tamoxifen, we can really get get the exon 3 cleaved away, okay? So what we did is to inject these mice, okay, and look at the exon 3 where it is excised. So we know that tamoxifen doesn't work immediately. It takes a few days, even weeks, to get around all the cells. So 14 days after the tamoxifen was injected, we looked at the kidney of a mouse, at the liver of a mouse, where we saw that Gne-Flag is here. Okay, again, if exon 3 is here, we have the flag. GNE. If exon 3 is excised, we have a knockout, so we will not have the flag either, okay? So we are looking here for GNE-Flag. So in kidney, we see the GNE-Flag. In liver, we see the GNE-Flag, and that's good, that's what we wanted. In muscles, in some muscles, we don't see GNE-Flag, and that's good, that's what we wanted. But in some muscles, we did. So we thought that 14 days post-tamoxifen was not enough. So we looked at another mouse 30 days after tamoxifen infection. And here indeed we see the GNE-Flag in kidney, GNE-Flag in liver, also in brain, but in all the muscles, quadriceps, gastrocnemius, tibialis of 2 different mice, we don't see any tamoxifen at all. So we were happy with that. And then when we saw that molecularly everything works, we grow a colony of homozygous mice that have the LOX GNE-Flag, and we injected them with tamoxifen. And the aim is, of course, now to follow up for muscle function, muscle force, and of course, histology of muscle. But then we thought that we don't have GNE in muscle, but we have GNE in all the rest of the organism. And if we have GNE in all the rest of the organism, we also have sialic acid in the rest of the organism. And maybe this sialic acid could be recycled from the serum glycoproteins and will get to the muscle. And then if the hypothesis that sialic acid is responsible for for muscle impairment, so then we will not be able to see that. Okay, what we know is in fact indeed that in liver, that liver is the main source of serum glycoproteins, and that those serum glycoproteins can release the sialic acid that is then uptaken by all the organs in the organism and also skeletal muscle. So what can we do to prevent sialic acid to get to skeletal muscle? So what we thought is if we knock out GNE in the liver, then we will be able to decrease dramatically all these glycoproteins in the serum so that nothing will reach the muscle. Okay, so how can we do that? So we found a virus, an AAV virus, that encodes for the CRE enzyme, right, or the recombinase enzyme. But this enzyme is now led by a liver-specific promoter. That means that the CRE recombinase will work only in the liver. Again, to remind you, in our mouse, we have the LOXP sequences in all the organisms, okay? So also in liver. So now if we inject these mice with this virus, okay, we will have to see that indeed GNE is knocked out in liver, okay? So that's what we did. We then monitored GNE-Flag expression in liver to see if it really happened. And indeed, we can see here in non-injected mouse, we can see GNE-Flag in liver and kidney. And 7 days after injection of this virus, we can see GNE-Flag in kidney, but we can see almost nothing in liver. Okay, so it's a dramatically decrease in liver. But again, we saw a little bit, so we were not happy with that. So we waited again for 30 days. And here we can see that indeed the liver is completely without any GNE there. So this works, but we were concerned that maybe GNE knockout in liver will not let the mice to develop well or to live even. But what we see is that 3 months after infection of the mice with this virus, the mice look completely normal and live normally. So this is a good system. And what we did in fact, is have a mouse that are double tissue GNE-Flag knockout. That is, that the knockout is also in muscle, as we first designed, okay, but also in liver. So we will have this GNE knockout both in muscle and in liver. We will then follow up for muscle function and histology. So we have those now just for a month. So it's not enough. We are beginning to do the muscle function assessment and evaluation, and hopefully we will see something happen in muscle. So here we have 1 month after the tamoxifen and the AAV CRE injection. We can see indeed that we have GNE-Flag in kidney, but nothing in liver, nothing in quadriceps. And we looked at other muscles, quadriceps, gastrocnemius, tibialis anterior in 2 different mice. We see just nothing, okay? So we are confident that this model will give us some clues about what's going on in GNE knockout muscle. So we want to characterize, of course, the motor performance the muscle force of these mice, and of course the histology when we sacrifice the mouse. And then we will do also a more omics analysis to try and elucidate the molecular processes that are really changed because of the lack of GNE. And of course, if this works and we have some kind of muscle pathology, we would be able to use those mice to evaluate the efficacy of any treatment. In summary, we have here a GNE knockout zebrafish that has been established and characterized. We have a GNE knockout only in muscle mouse, and we have also a GNE knockout in muscle and in liver. So hopefully these new tools will allow us a better understanding of GNE potential functions in muscle. We will be able to identify novel biomarkers and to assess any therapy treatments. So when we talk about therapy for GNE myopathy, we all know that NDF is committed to the development of gene therapy, as I am also personally. However, we have to be aware that other treatments could be efficient as well. For example, drug therapy. We all remember the Ultragenyx trial with sialic acid. And there is also a trial at NIH with the sialic acid precursor MANAC. And here, what I want to you to be aware of is the efforts developed in Europe in drug therapy for GNE myopathy. There is in Europe a research program for rare diseases. There was a call in 2020 for preclinical research to develop effective therapies for rare diseases. So for this purpose, a transnational consortium was established, and they call it ProDGne, which presented and got funded, in fact, for the project, novel therapies approaches to target GNE myopathy. So what do this consortium want to do? We know that one problem with the sialic acid and MANAC is the difficulties of these molecules to enter the organism, the cells. Therefore, the aim of this project is to increase the permeability of MANAC by chemical modifications. So the consortium is a unique collaboration between Europe and Canada, and the expertise is in clinical genomyopathy and in basic research, mostly in glycomics and in glycobiology, and specifically in sialic acid. The developed prodrug, when processed within the cells, becomes active and highly increased sialic acid in the cells, and is also more stable. So who are the people behind this consortium? So first of all, this is Michela Onali. I guess most of you know Michela. Michela is the representative of the GNE Myopathy organization in Italy. And she is, in fact, the person who organized and contacted everybody else and convinced them to apply for the grant. And indeed, she did well because they got it. Then we have Paula Videira in Portugal. She's a glycobiologist, and she is the PI or the responsible of the entire group. Then Fabrizio Pertusati, he's an Italian researcher that works in the UK, and he is in fact the chemist that is modifying the MANAC molecule. Hans Rochmüller, he's a clinician. I guess most of you know him also. And Rudiger, that is also in the SAC of NDF. he is an expert of sialic acid, so he joined this effort too. And Dr. Caboni from Italy is also a glycobiologist. So these people all together have been working for a year already and have made some progress. They showed that the modified MANAC indeed has an increased permeability to cells, and this leads to an increased production of sialic acid in those cells. So now they are continuing by testing this molecule in patient cells, and of course they are looking for animal models to test this efficacy. So here we can also help with the models we have now. We are now developing, and here I want again to talk about the zebrafish or the muscle knockout models in mouse that hopefully will be ready very soon. So here I want to thank all my team for the fantastic work they have been doing for the last years. And also our long life collaborator in the clinic, Zohar Argov. And of course, the NDF for its long and lasting support during all these years. And also the IFM Teleton and the Israel Science Foundation that have supported part of these studies. So thank you very much for your being here, and here are the takeaway messages. I hope you can get home. Thank you.
Thank you very much for that. Greatly appreciate that excellent lecture. Very interesting on several different preclinical models for GNE myopathy, and obviously great utility for that in all the different areas in which you discussed. So if anyone has any questions, it would be great to put them in the chat box, and I can read those out. The chat box you can access at the bottom of your screen, and happy to do that. Actually, we already have a couple of questions coming up in the question and answer section. Let me start with a question. Did the mouse model developed so far show any muscle phenotype?
Well, that's the million-dollar question. Again, what I pointed out is that we are now very close to the tamoxifen and the virus injection. It's a month or two, so it's a little bit early, I guess. After 1 month, we didn't see any motor function defect.
Right. Yeah. I mean, that's at least
but
initial
we didn't
indication.
sacrifice any mouse yet.
Right. Yeah. I mean, given the adult onset, given the adult onset nature, that makes a lot of sense.
Yeah. So this is not surprising, let's say.
Certainly. There's another question in the Q&A section. Thank you for an absolutely fascinating presentation, and then asking a clarification. If they understood correctly that the phenotype in GNE knockout zebrafish showed heart muscle impairment. Is this different from humans where the heart muscle does not seem to be implicated in GNE patients?
Yeah, that's right. But here again, we have a GNE knockout. Okay, you have to be aware that there is no GNE at all. Okay, so we expected to have a more dramatic phenotype. That's why we did it. That was our strategy, to use a GNE knockout, complete knockout, so we can really emphasize the function of the muscle of the GNE, so we can see a more severe phenotype. Okay, so again, it could be that GNE mutation in zebrafish could also lead to a heart phenotype, but I don't know. But anyway, here again we have completely absence of GNE. That's not what happens in human patients, where we have GNE, even normal levels of GNE. Mutated, but normal, but as far as quantity, we have the same levels of protein.
Certainly. And there's actually a follow-up question there too. You said there's no antibody to detect the lack of GNE in the mouse model and hence the presence of the Gne-Flag on the mouse. Does the research on lectin staining help in this case?
Well, lectin will detect sialic acid, sugars as a whole, okay? Here we want to see if we have the GNE protein at the first level to know if the system works, okay? The system was designed to knock out GNE. So we wanted to see if indeed we have knocked out GNE, okay? Before we look at sialic acid or whatever, yeah. So these are two different targets. Okay? Lectins target sugars or sialic acid specifically for our sake. And this flag antibody targets the GNE protein itself. Did that answer the question?
If not, people can follow up. Yes, they answered both questions. Thank you. Are there, if there's any other questions, please feel free to put them into either the Q&A or the chat box. I actually had a question as it relates to the using the zebrafish model for screening metabolites or portions of the metabolic pathway and substrates. So is that as straightforward as just supplementing it in the water there? Is that absorbed and metabolized effectively by the fish when that takes place? Or I don't do a lot with fish, so I'm just kind of curious how well that works and if you have to have extremely high levels of it to actually get it to be absorbed.
Well, I am not an expert on that either, but as far as I understand, most of the metabolites that are used for that are indeed added into the water and they get into the fish spontaneously. So that's right thing, yeah.
That makes it a little easier than some ways to screen that. So that does make that a very useful one.
Of course.
Actually, so is yeah, please go ahead.
No, I say maybe not everything you want to test. I mean, if we want to add GNE, I would say the GNE protein will, I'm not sure, I don't know if it will get in or not. But we are talking about many different metabolites that are used for screenings of many things.
Right.
So those are added into the water. Yeah. Now for GNE, we can use the AAV, of course, and see if they really can infect the fish.
Thank you. Are there any other questions? Oh, yes, I see one there. How do you explain GNE expression in some muscle types? Was the CRE not expressed in those muscle types?
Okay, what I showed is that we have some remainings of GNE in some of the muscles. I don't think it's related to the different type of muscle. It's not that we saw in all the quadriceps more GNE than the others. It was completely random. And I think, yes, it is because tamoxifen didn't reach all the muscles at that time. That's why we waited for a longer time and then we saw everything was fine. So this, I would say, is the availability of tamoxifen is time depending, and it takes some time, depends where you make the first injection, if it's close to some other muscles, okay? So I guess the ones that are closer to the injection site will show the GNE knockout, the effect earlier, okay? But this is intuition, I don't know. It takes time to the tamoxifen to get absorbed. We inject it subcutaneously. So it takes time to it absorb and get into the whole body. That's why we have to wait for a month to be sure we have a total GNE knockout where we want to.
Exactly. Well, let's give just one more second, see if there's any more questions. While that's going on, I will remind the audience that we actually have two events in the lecture series coming up in the next month. So two opportunities to have an excellent presentation on GNE myopathy and different efforts, both for treatment and for how to effectively live with GNE myopathy. That's sort of the topic of our first one, which will be on July 17th, again at 9 a.m. Pacific time. And that is on mindful living, an alternative approach to living and fulfilling life with GNE myopathy. And that is by Dr. Talibian, who will be presenting. And then on the 31st of July, we will also have another lecture. We were just talking about him from Merger on the post-translational modification of GNE, which should also be an interesting talk as well. I hope people can attend that. I don't see any additional questions coming online, but there's several people listing thanks in the chat box, and so I will thank you as well for this excellent presentation. Greatly appreciated. Thank you very much.
Let me
And Jeffrey, Jeffrey, please go ahead.
Jump in. A couple of things I wanted to say. We continue our collaboration with others. For example, the European group. I was talking to Michela in Italy on Friday morning. So we're continuing discussions and ensuring that NDF is part of every collaboration that's going on. I really want to thank you both for the time you spent this morning. I want to remind everybody that the NDF has funded the research that Stella has been doing. And obviously funding of research only comes from dollars raised. So I want to throw in a plug for our gala that is scheduled to take place in November, at which Dr. Mitrani-Rosenbaum will receive the Scientific Achievement Award. And we're very delighted to be able to give her that award. And hopefully by November, she will either be able to travel to Los Angeles to receive it, or we will be honored and delighted to hand it to her remotely. So let me thank everybody for their attendance. I thought this was an excellent session. I learned a lot, and I'm a lawyer, not a scientist, but everything that I learn moves us forward. So I really want to thank you both, and thank everybody for participating from around the world, and look forward to seeing and hearing from you in the weeks and months ahead. Thank you so much.